Hey everybody, Professor Gassimi here. First of all, hope you all had a wonderful spring break. The purpose of this week's lecture is not really to cover any new content, but is to put something like a capstone on all the content we've covered to date. The reason we're doing this is because you have at this point in the class actually obtained exposure through the lectures as well as the assignments to all of the core components you will need to build a full stack web application on your own. You understand JavaScript, HTML, and CSS, how those control the front end. You also understand things about the back end, how to design a web application in Flask, what sessions are, what cookies are, and so on, as well as how you can use databases to power your web application and how the intersection of those three components, the databases, the front end, and the back end, can make really powerful web applications. So the best way to bring that together, I think, is for me to show you an actual example of an application that I actually personally started building last year. This was built in collaboration with the National Institutes of Health, where I served as a Data and Technology Advancement National Service Scholar last year. I'd like to give you an overview of the tool that I built because it actually uses all the same things we've learned about in class. This tool uses MySQL as the database, it uses Flask as the backend, and there is JavaScript, HTML, and CSS for the front end. So in this lecture, I'm going to first take you through the application that was built so that you have an idea of the kinds of things that can be built using the tools we've covered. And then I'm actually going to show you on the web where you can access the open source code repository for this tool. And we're going to go through some of the components together. So let's get started. You all already know who I am. And so I'll skip that part. These slides that I'm showing you, by the way, are from the work that I did while I was at the National Institutes of Health last year. And one of the things that I highlighted to the folks at the National Institutes of Health, but I also think is worth highlighting to all of you today, is that our laboratory is interested in developing tools that use artificial intelligence, but also have a human-facing component. And that's the reason I teach the web design course, is because if you want to build tools that talk to people, interact with people, you want to think about packaging that within usually a web application of some kind. So if this is an area of interest to you, actually, I'd love for you to reach out, especially now that we're getting close to the end of the semester, to see if there's some research that we can do together, either in the lab or if there are internship opportunities I can help connect you to. We've had other alumni in the course actually come out of the class and work on developing some pretty impressive web applications, including parts of what you're going to see today. Okay, so let me give you an introduction to this project that we worked on, this full stack web application. Last year when I started at the National Institutes of Health, I was working with some really sharp people in the BRAIN Initiative. And BRAIN Initiative is a national project, is the way to think about it, that is trying to promote a dynamic understanding of the human brain, which is this mysterious, unknown scientific object. We understand pieces of it, but the whole system is still very mysterious. And when I came on last year, that text that you see in the center of the screen is what they asked me to work on. They said, Dr. Gassimi, we want you to deliver a functional proof of concept of the BRAIN Initiative's workspace to organize the knowledge space, or BrainWorks for short. And they wanted this tool to help them discover more comprehensive theories of brain function through knowledge integration. Now, I'm showing you this statement of work, if you will, because when you all graduate, and as seniors you all will very soon, when you graduate and go out into the workforce, it's very common to receive very high-level missions, sort of like what you see here, and to try to think creatively about how you can build applications, tools, generate algorithms, and so on, to solve against some of these abstract statements. That's something that people really value in the workforce. So what we tried to do to start with was break the problem down and try to understand how we could use tools in computer science, data science, and web application development to try to solve against that mission statement. And the first thing we started with was this observation that brain data is very heterogeneous. So if you want to understand the brain, there's a bunch of contextual information you also have to understand that all informs, your understanding of the brain. There's genetic information, right? So what our genes say impact things about how our brains operate. There's cellular level information you have to understand. So what are the individual cells doing? You have to think of the brain as an entire system, so at the neurological resolution. There's a physiological context, the brain within the greater context of the body. For instance, it will function differently if you're feeling very stressed versus if you're not feeling stressed at all. Then, of course, there's a behavioral context. So the way your physiological system and your neurological system will behave depend on, if you're taking a jog, if you're sitting with friends laughing, and so on. And then finally, there is a greater environmental context that impacts everything else, including your behavior, your physiology, and so on, such as if the environment is highly polluted, you might behave slightly differently, not only physiologically, but also behaviorally, than if you were in a place that was very clean. The thought here was that if you want to understand the brain, which remember was the mission of the project, you would need to integrate these modalities together. You need to understand the environment, the genomic, the cellular, the neurologic, the physiologic, and the behavioral all together in a complete context to empower brain research. So something like this, pulling all of those together. The thought was that would be really challenging for even the most gifted, brilliant scientist to do alone. And that's where tools in data science and computer science more generally could be helpful here. Okay? The way we ended up approaching the problem was through something that I call a value-centered approach to data science. And we're going to touch on as we go through this the piece that is actually very related to web application development. It's actually a huge piece of this. But first, let's talk through the diagram. You can see at the center of the screen here, we've got something, a value. It's the intersection in this Venn diagram. Now, value here is something that people want to achieve with a piece of technology. In the case of the Brain Initiative, they wanted to be able to better understand the brain, right? They wanted to unite information from all of those different domains, neurologic, behavioral, environmental, etc., so that they could more easily understand the knowledge surrounding the brain. Well, if you want to accomplish this value, and you're going to use a data science methodology to accomplish it, there's always three things, specific outcomes, I should say, that you will use to achieve that value. There's automation. So that's this intersection up here. And that's the use of algorithms to convert low-value information into some high-value information. There's also augmentation here, which is the combination of human and machine capabilities to create more value than either the human could alone or the machine could alone. And finally, there's discovery, which is the process of communicating information so that people can extract value from it on their own. Now notice that this Venn diagram, at the periphery, we have three circles. We have information, algorithms, and communication. Let's define those three, and then we're going to speak about where web application development is critical to accomplish this value in the center. Information is related to data. It's not exactly data. It's sort of the cleaned, pre-processed, easy-to-use version of data. Think if you had a square table, it was already pre-processed, it was good to go. That's what we mean by information. This supports your discovery and your automation data science outcomes. On the algorithm side, these are tools that are used to predict one kind of information from another. Algorithms support the automation and the augmentation data science outcomes. And then, of course, there's communications. And these are methods for representing complex information in simple forms. These are what empower the discovery as well as the augmentation outcomes within data science. And it's at the nexus of these three, when you can provide automation when required, augmentation when required, and discovery when required, that you achieve ultimately value through data science. Now, web application development is an absolute must absolute must if you want to do the discovery, the augmentation, and the communications piece of this. There's no way around it in most cases. And so a pillar of actually, I would say, being a good data scientist, really an elite kind of data science talent, is not only to understand, deep learning and data systems and so on, but to master the kinds of skills that we've been going through in this course. And I hope to demonstrate that to you as we continue to go through this lecture today. So the project that we worked on last year proceeded in 5 phases over 12 months. You can see on the far left there, I've got something called a specification phase, and I've got that little Venn diagram that we just discussed. That specification phase basically is trying to articulate exactly what this value is. This is going from that statement we saw on the first slide, kind of the high level, a little bit abstract statement of what we wanted to accomplish, to a very specific instantiation of our goals, what we specifically want to accomplish and deliver by the time we get to the end of the year. Okay, that proceeds to a phase where we focus most of our attention. You can see the red triangle here. Most of our attention is on information, trying to collect data, organize data. We move into a third phase that is still working on some of the information, as you can see by the red area here, but we shift our focus a little bit towards the algorithms and communications workflow. So writing natural language processing models, deep learning models, and so on, and also starting to really focus on building the web applications to enable the, as you can see here, the discovery framework as well as the automation framework. Finally, we get to the augmentation phase, which is where we sort of finished with the pre-processing of the data. We've got a preliminary version of our web application, our algorithms done, and we're sort of knitting everything together into a single framework so that finally in the 5th stage, we can deliver the value that we were aiming for back here in the specification phase. So that's sort of how the project flows. So let's talk about each of these in the course of the lecture. The first one was a requirements capturing exercise that we did. And effectively, to capture those requirements, whether you do this in your own industry job later, or you go on to become a scientist, this is an important part of any project that you do. You'll speak to a set of stakeholders, as I did last year. These are people both within National Institutes of Health, as well as preeminent scientists outside of the National Institutes of Health. And we asked them just very directly, hey, how can we help empower you to get better understanding of the brain, of neuroscience? What are ways that data science can help? And we had some open and honest conversations with them. And here's what we arrived at as the answer to the question. They said, in the first order sense, if you could develop a tool for us that would take scientific papers, things you pull up on Google Scholar and such, and would turn all those scientific papers into a knowledge graph of some kind, a representation of that, that would be really valuable. It would allow us to explore the scientific literature. It would allow us to integrate the findings across papers. It would just make our life so much easier. So what we decided the specific goal of the year would be was to build a web application, actually, the stuff we're talking about in this course. And that web application was going to use AI, as well as obviously all the things that we've discussed to date in the class, to organize the neuroscience literature and the broader scientific literature, in fact, as an intuitive and interactive knowledge graph. To give you a concrete example, look at the image on the bottom left there. You can see I've got each of those 6 kind of areas we spoke about previously, the things that inform research on the brain. And there's papers that are written on each of those domains, right? There's papers written on social aspects that impact brain and brain function, genetic aspects, and so on. What we'd like to be able to do is take those papers and understand the core conceptual objects within them: stress, age, cognitive ability, dendritic branching as an example, as well as the specific relationships that are espoused within those papers. So, for example, dendritic branching impacts cognitive ability. We'd like to be able to turn a collection of papers into something like that graph that you see there, and that's the objective. Now, why would we care to do this? Why were people interested? Well, because neuroscience, just like many domains of science now, the knowledge is distributed across a very complex and rapidly evolving scientific landscape that even the most clever person would find it challenging to master. What I'm showing you here on the right-hand side is a graph, and you can see I've got the year along the x-axis, and I've got the number of papers related to neuro-related topics along the y-axis. And what I'm showing you here is a couple of call-outs at different points in time. Here around 1970 roughly, if you wanted to keep up with all neuro-related articles, you'd need to read something on the order of 13 papers a day. That's a lot of papers. I mean, they can be 10 pages, they can be 20 pages, but even back in 1970, you'd have to spend a whole lot of time if you wanted to keep up to date with every paper that was being published on the articles. Even if you only took the upper half, let's say you had a way of sorting them, you'd still have to read 6 or 7 papers a day, which is a lot. But if you fast forward to closer to today, around 2010, 2015, we're up to 70 papers a day on the topic. It's because there's a lot more scientists today and a lot more people creating science. And so even if you were to take the top quarter of this, if you were to, forget it, let's say you were only to take the top 10 of these, you'd still have a hard time keeping up with all the literature. There's a lot going on. And so the motivation here is, can we, by turning those papers into a knowledge graph, can we simplify the process of trying to understand the literature and where it's going? That was the goal. Okay, so we understood the goal, right? That was that first step. On the left, the specification phase, we call it. We then moved to the second step, which is these data to information phase. And that focuses on collecting data to enable the platform realization. The place where we got that data from was 3 government resources. The first is the report tool, which stands for the Research Portfolio Online Reporting Tool. This basically provides access to reports, data, analysis of NIH research activities, and the funded projects, as well as their affiliated publications and their patents. So the way to think about this is that the National Institutes of Health, the reason it's called Institutes of Health is because there's many institutes within it. And I'm showing here along the x-axis in the chart to the right-hand side, each of those institutes and the number of what's called projects within them. Those are grants that they've given to scientific investigators to answer interesting questions like, hey, tell us how COVID-19 propagates. Go do some interesting research to find the answer to that question that we don't know yet. Or, hey, tell us how we can use CRISPR to perform gene editing more effectively, and so on and so forth. Okay, so there's a bunch of these active projects. The first thing we did is we went and we collected all the information about the existing active projects that are occurring within the National Institutes of Health, not only today, but from 1980 until today. Over a long time frame, we collected it. Now, typically what happens within scientific institutions, and universities, in fact, are part of this ecosystem. Your professors are always doing research, right? They're always working on papers, publishing stuff, flying off to conferences, and so on. That's because when those projects, those awards are given by places like the National Institutes of Health, oftentimes they're given to professors like me or like other professors you have in the CS department and so on. And those professors take those awards, they do some science, they do an investigation, and they publish a paper to divulge what it is that they found when they did the investigation so that people can read it, learn, and knowledge gets advanced. Well, those papers that are tied to those awards are also things that we collected here, right? That's where we're going to be able to extract that knowledge graph I spoke about. Specifically, we went to a service called pubmed.gov, and we extract, which contains something on the order of 32 million citations from life science journals, books, and other sources of the biomedical literature. And we said, hey, for every for every project we saw that was funded by NIH from 1980 until 2020, let's go and extract all the papers that were associated or the outcomes of those projects. And I'm showing you on the right-hand side here an example of work that was done on neuroplasticity as an example. So the height of this bar is the number of papers that was published on neuroplasticity. And I'm showing one example of a paper that was published in 2017 covering the topic of what is neuroplasticity. That's the kind of information we collected from PubMed. Okay, finally, when you get access to those papers and we know which grants funded them and the whole ecosystem around it, We also want to know things about the topics of those papers. Was this work on gene editing? Was this work on COVID-19? And if it was work on COVID-19, what aspect on its effect on respiratory systems and so on? The way that we collected that topical information for each of the papers was through something called the medical subject headings. And it's effectively a hierarchically organized thesaurus of continually reviewed and updated medical concepts. So the way to think about it is like this. For each of the papers that we collect, we also go and we give them a topic number here. So if we had, for example, A11.671.137, this exists in a hierarchical ontology here of concepts. And so I know that axons, based on the way that the labeling is done, is a part of neurons, and that mossy fibers, hippocampal, are a subcomponent of axons, and so on and so forth. So ultimately, the data set that we collected, which, by the way, a large component of which was stored in a MySQL database, the kind of thing you're using in the class, what we ended up with was this set of NIH-funded projects, 2.7 million of them actually, that accounted for $500 billion, that's trillion with a T, $500 billion in government investment over this 40-year period. It's a lot of money. And this $500 billion in investment to answer interesting questions went to something on the order of 5 million scientists who were affiliated with 100,000 institutions globally. These scientists authored something on the order of 4 million papers in that time period, according to the assets within our database. And when people write papers, just like you guys write your papers, they cite other papers as evidence that they're doing their work correctly, they're building on the knowledge base, and so on. So we not only collected these papers that the scientists generated from the NIH projects, but we also collected everything that those papers cited. So I basically went to the bibliography and grabbed anything in there that was also in PubMed. Okay, we mapped obviously from there to the topics that I mentioned earlier, the medical subject headings. And we also, just for fun, actually collected any patents that were granted to institutions that were a consequence of those projects. So what you're looking at here from a data perspective is the complete picture of the scientific universe from, at least within NIH's scientific universe, from 1980 to 2020. The grants, the scientists, the institutions, the papers, the topics, the patents, all of it. The information that we collected was through a pipeline that I'm showing you here. We effectively built some software tools that allow us to generate date ranges and search terms, come to select a subset of the literature, extract metadata and raw documents, and to process those documents using techniques in natural language processing, which is the other class that I teach here at Michigan State, to store them finally in a relational database in a triple store. Now what I'd like to do, I mentioned this natural language processing piece. I'd like to take you through that NLP engine that we built. So ultimately, let's take a step back and think about what the challenge is here. You have a bunch of papers, right? You have a bunch of scientific documents, and you can't just use those scientific documents alone to generate a graph. You've got to process them first, right? You've got to turn them into something that you can visualize as nodes and as edges. Okay, so the NLP pipeline that I'm about to take you through, that's what it does. It takes a document in on the one side, it does some processing, and it generates on the output a graph, some nodes and some edges. So let's explore how the natural language processing pipeline that we built in BrainWorks accomplishes that by means of a couple of examples. What I've got here on the top is the particular pipeline, and I'm going to run through this, each time highlighting in red what's happening. And we're going to do this natural language processing pipeline on some example sentences that you see here in the middle. Mary had a little lamb, his fleece was black as coal. She had it last year. Dolly was a female Flemish Dorset sheep, and so on. So let's get started. The first step in this natural language processing pipeline is to take the text content and to do something called tokenization. Tokenization, pretty simply, is taking tokens, which are sort of the joined sequences of characters without spaces between them, and figuring out how to separate those out so that you have objects that are semantically distinct with spaces between them. That is to say that year period, semantically what that means is year and I finished my thought, ergo the period. And so the first step in the natural language processing pipeline is to separate those things out so that you can interpret year as one concept and the period as I am ending my discussion of this concept. She had it last year. The next thing in the pipeline is something called parts of speech tagging. And this is what you did in your grade school, right? You went and you took sentences and maybe your grammar teacher told you you need to highlight which of the words in this sentence are adjectives, which are nouns, which are numbers, which are pronouns, which are prepositions, and so on. And what I'm showing you here is an example of the natural language processing pipeline's ability to do that highlighting. Once you've done that, the next step is something called lemmatization, which is to figure out that it's going to be easier to do predictions with this language or to represent this language if we simplify it. So some of the simplification might be, let's jump back and forth. We had, she had it last year. We might simplify it because have and had are related in terms of what they're trying to communicate. We might simplify everywhere we see had to saying have, just so that we make the vocabulary in this smaller. We might also come and say, instead of Dolly was a female Flemish Dorset sheep, we might replace the word was with be, so that once again, we're just reducing the total size, the total number of unique words in here, and it's easier to understand that they mean the same thing. Okay? That's what lemmatization is. The next step in the pipeline is something called dependency parsing. And what this does is it takes words that are semantically related and it groups them so that they're treated as one concept. For example, if you're reading the sentence, Mary had a little lamb whose fleece was black as coal, when you don't think of a and little as being distinct, you think of a little lamb sort of like it's one word, kind of in your mind's eye. As in, the a and little are very tightly related because they're descriptors of the lamb. Okay, and the same as who applies to whose and fleece. So the process of dependency parsing in NLP is sort of treating these as one semantic object. You sort of are bringing them together so that you interpret them together. The next step is to perform something called coreference resolution, where you take the text, for example, like you see on the first sentence, Mary had a little lamb whose fleece be black as coal, and to say, maybe if we jump back, the she in the second sentence is in fact Mary from the first. And maybe the it in the second sentence is in fact the little lamb. And to perform that mapping across sets of sentences for representational purposes. Now, once you have the data in this state, you perform something that's called triple extraction. And triple extraction is taking those sentences and breaking them into this table that you see on the left that's called this semantic triple. It's a semantic triple because it has what the subject is, Mary, it has a relation, had, and it has an object, a little lamb. And why is this representation something that we were interested in ultimately with this pipeline? Because remember, we put a document in on the one side, and now we're getting these triples out on the other side. The reason we care about this is because you can represent this as a graph, right? Mary could be a node. Dolly could be a node. Clone could be a node. And the edges could be, in, last year have, had, and so on. So we've turned the document into a representation where we can visualize it as a graph now. And that was the objective, if you recall from the start. But of course, after you've done this triple extraction, there's going to be a lot of subject-object relation triples in this data, and we need a way to thin this out. Basically, we need a way to identify some of these that may not be very relevant. Let's say it's the ones in red that are not relevant to what we want to visualize. The way that we did that was by basically saying, hey, we're only going to keep the subset of these where we see a concept that shows up from the Unified Medical Language System, or UMLS. UMLS is basically, it's an integration of distinct key terminologies, classification, and coding standards that promotes the creation of more interpretable biomedical information systems. I won't take you through UMLS, but just making you aware of it in case you want to use it for simplifying language or doing things in a project of your own. Ultimately, what we end up with, though, after looking for the existence of those terms, is a much smaller subset that is supposed to represent what really matters in the paper from a graph perspective, as we're showing here. Okay, so we've taken you through the specification. That was, what problem do we want to solve? We took you through the data that we collected that we put in that MySQL database, remember? Then we took you through the generation of some algorithms and communication. And by the way, all that was written in Python, okay? The same language that we're using to support the web application. And then finally, there's this augmentation that we took you through, or we're going to take you through rather right now, which is going to talk about how we generated the dynamic graphs using web applications and supporting frameworks. So the first thing I'd like to show you here is a couple of visualizations of graphs that we generated using the tool. And then at the end, I'm going to actually take you to the live version of the system and show you some examples of the live tool as well that's running on the World Wide Web today. So what I'm showing you here is a publication topic graph for everybody who received an award from the BRAIN Initiative. And there's nodes and there's edges here. Let me take you through what those mean. A node here is a topic that some scientists published on between 2014 and 2020. And the larger the node is, the more scientists did work on that domain. So you can see, for example, magnetic resonance imaging is really big. The text above it, at least, is really big. That means there was a lot of folks who, when they were doing their research, the topic of magnetic resonance imaging showed up in their papers. The same is true of optogenetics and single-cell analysis. Notice these three things have in common that they're all kind of methods, right? They're things that people use to study other things. And appropriately, there are edges, therefore, between these topics, like optogenetics or MRI, and other topics that show up. What this edge means is that two topics in a paper co-occurred, because a paper is not usually about just one topic. You're not, for example, typically just studying magnetic resonance imaging, but you might be studying magnetic resonance imaging in the cerebral cortex, which you can see there's a connection to. You might be studying magnetic resonance imaging for brain mapping, which is why there's an edge here. You might notice that these edges have different thickness, and that's because the thickness here represents something about the co-occurrence. So for example, the visual cortex has a pretty thick edge with photic stimulation, whereas neural pathways has a relatively thinner edge with movement, which means that the co-occurrence of topics between visual cortex and photic stimulation was higher than between neural pathways and movement. So this is an example of the kinds of things when we had the data in shape that we were able to visualize. It's getting one step closer to that web application that people can touch. Let's look at the kinds of things that we can also investigate using the data and the visualization engine. If we take one of the topics here, like neuroplasticity, we can look at the co-occurrence of topics in publications to understand trends in research, right? That is going to help answer the first question we were going after. Where is science going? What is understood about the brain? How has it changed? So what I'm showing you here is a plot with two axes. Let me explain what the axes mean, and then we're going to talk through how to interpret this. Remember how I told you the data was collected from 1980 to 2020? Well, that's our x-axis here. All the way on the left is 1980, all the way on the right is 2020. And then I've got here along the y-axis the number of papers that were published in hundreds. So zeros down here, 800s at the top for each of those years. A higher bar means more papers were published that year on neuroplasticity. A lower bar means less papers were published. You might also notice that there's two kinds of bars. There's an orange bar and there's a blue bar. The blue bar means that, hey, the research that was done in, for example, 1984, you can see here, a very small fraction of it was in humans relative to the fraction in animals, which is done in orange. As time went on, you glaze your eyes from left to right, you can see there's a trend, right? People started to use more animals in the course of research more humans in the course of research proportionally than they did animals compared to the earlier times. But because the data here is hierarchically organized in terms of the topics, there's some interesting opportunities that you get for visualization here that you could do embed in a web application as an example. You can take that animals topic that you see, and animals describes a whole bunch of things: birds, sheep, rabbits, etc. Well, we can break it out and we can say what were the specific animals that were part of that orange bar in the previous slide. And you get a very interesting or a more nuanced kind of story about research in neuroplasticity now. What you can see is that in the earlier times, when people were studying neuroplasticity, they were using rats predominantly. That's the green bar here, right? Look at the proportion of the green relative to everything else. And as time went on, there was a shift away from the use of rats toward the use of mice. That's the orange bar that you see here. But you can get even more detailed, again, because the data is hierarchically structured. You can even ask what kinds of strain of rats were used in the course of the research. And you can watch how trends in the use of specific kinds of rats went up and went down over time. Okay, so what have we gone through? We've gone through the specification. We've gone through the data that was used. We've gone through the algorithms that were generated. We've gone through the user interface that was developed, or at least what the user interface could do. The last question we were interested in was tying the loop and showing the value. Remember, like, you always want to come back and tie this piece, the value, based on everything that was built to support it. So let's talk about a couple of things we did that I think were pretty cool using the tool. We did several of them, actually, several use case explorations to validate the value of this tool, but I'm only going to show you a couple of them. Let's start with what I think one of the really interesting ones was to start with on how collaborative projects impact author networks and papers. So what we did is we went to the tool and we said, hey, let's go take 30 scientific labs that received an award that was supposed to help them collaborate in 2017. Let's call that our intervention group. Let's go take 30 other labs who didn't receive that award, but we know that they're very productive. They get lots of grants. The government really trusts them to do good quality research. So because they're receiving all these grants, if they wanted to, they could probably collaborate with anyone, right? They have lots of cash. They're ready to go. Well, let's go look and compare those two groups. Let's look at them 4 years before the first group received their grant to help them collaborate, and compare that to 4 years after, because we want to see if that grant that was supposed to increase collaborations, what it did when we compare the two groups. Okay, so we're going to look at the change in publication output and collaborations for that intervention and the control group. So what we did is we generated a plot like what you see here on the right-hand side that illustrates how that collaborative structure changed between the scientists. This is a plot from the random control group. And specifically, I want to point out how you interpret this chart. You can see here we've got nodes and we've got edges, just like we had before. The nodes in this case are individual people. So this orange node, that's a person who is in a scientific lab. Like, for example, if you joined my lab and you were doing science and you became a researcher, you'd be a node in this graph. Okay, and when you have an edge between the nodes, it means that those two nodes over the 8-year contrast period, they worked together more closely. They published more papers together. They did more projects together. That's what an edge between them means. You can see there's blue edges and there's yellow edges. Well, a blue edge means that two people in the same group, i.e. the same colored node, that they increased their efforts together. A yellow edge means that two people from different groups started doing more work together. So when you glance at this with your eyes, what you see is that people do collaborate across groups in that control group that we looked at. The people who did not receive the grant to help them collaborate more, they still work together across groups, but for the most part, they were working within their own group, right? The blues are sort of clustered together. The oranges are clustered together. You know, each of these kind of colors are all kind of clustered on their own, although there are edges across them. Now, when you compare this against the intervention group. Those are the people who received this award that was specifically supposed to help them figure out how to collaborate more effectively. Look at the difference here. I'll pan back and forth, and you can see it with your eyes. Here's the random control. Here's the intervention. Here's the random control, and here's the intervention. It's a world of difference, right? This group, if this were a room of people and you were looking from the top down, because each of those nodes is a person, and I said, which of the two groups, this one or this one, is better integrated, and you were looking top down at them, obviously it's this one, right? In fact, if you take quantitative measures of the change in collaboration, you get a 2x increase here compared to the previous one. So huge world of difference that you're able to see and visualize, not only because we had the data available, but because we built a user interface around it, hosted through a web application. We were able to run some very interesting statistics and surface those statistics on how these collaborative projects impacted the author networks and papers, comparing the control and the intervention group. In this case, I'm showing the average number of collaborators per scientific project. You can see for the intervention group, it went from 6.5 to 9.2. That's a pretty big jump compared to this control group, where it went from 6.3 to only 7.4. We were also, because in the data set, we have not only information about who those authors are and what their papers were, but we know the topics, remember? So we can look at what kinds of work happened at topical intersections, like you see here. More work for the intervention group happened at the topic intersections than it did for the control group, which we thought was pretty interesting. So we then started to ask, well, can we predict future graph edges via graph dynamics? Is that possible? And let me try to motivate why we cared about this. Here's that intervention group, but instead of it being the 4-year contrast, so 4 years before and 4 years after they received the award to help them collaborate, here's what they looked like 2 years in. So they've not had all the time yet to sort of forge those collaborations, to make meaningful relationships. They've only had 2 years, they're only 2 years into the project together. Well, what do you notice here? You notice that there are connections between people, but there's also these islands. Look at this green bubble on the left. This is a lab that's working together, but they're not really collaborating with other people yet. You've also got this island here in the middle with the greenish colored nodes that are kind of working together, but they've not merged in with the greater graph. Now, when you go from a 2-year contrast to a 4-year contrast, here it is again, a 2-year contrast versus a 4-year contrast, you can see that that bubble gets pulled in, right? This green bubble is no longer floating off to the left. So it means that if we just give them a couple of years, we're able to see that these people do in fact start working together. They start collaborating, and we were able to measure that. But here's the thing. Notice that what's tethering this green node to the larger network are these two, let's call them heroic green nodes here. And they're attached to some other elements within this network. So there's not that many points or people that are connecting the scientific initiatives of these laboratories. And the question was, could you predict ahead of time that this node here in the green bubble, 2 years in, was going to collaborate with this node on the other part of the network to draw it in? Because if you could have identified that sooner, maybe you could have introduced them ahead of time, and maybe they would have collaborated sooner and the networks would have been merged sooner. That's the idea. Okay, so we tried to explore the extent to which this might be possible. And we did this by trying to predict future graph edges via graph dynamics. More specifically, we took 5 years of data. So we went to 2015 through 2018, and we said, hey, let's study how these authors of the papers, the scientists, were connected to each other. And then let's see if we can predict who's going to work together in 2019, so 1 year later. The way we did that is through a technology called Node-to-Vec, or specifically, in our case, Dynagraph-to-Vec, which is a machine learning technology that takes something like a graph that you see here on the left and transforms each of the points in the graph to a point in a vector space, like you see here on the right. Okay, and we essentially were able to take the 5 years of data, here's 2015, 2016, 2017, 2018, and the connectivity structure in each of the graphs, and turn it into a movie of the points in that embedding space that you see on the right-hand side. Now, why does this matter? Well, because if I know 2015, 2016, 2017, 2018, and I see those dots on the right-hand side moving, they seem to have a trajectory, right? They have like a velocity, they have an acceleration, they have a position. Well, I can then predict where they're going to be in the next time point based on that movement, the same way you could with if you were rolling a toy car on a table, and try to make predictions about which nodes in the future might be connected. And we actually tested this and it worked pretty well, but it's a project we're still undergoing in my scientific research group. So if this is the kind of thing you're interested in, please do reach out to me. I'd love to chat with you. What I'd like to do now is switch gears, and I'd like to actually take you through the actual web application itself for Brainworks. So give me a second while I just quickly pull that up here. Okay, let's come to this one. So what you should be able to see shared on my screen now is the actual Brainworks application. You can see I've got my browser open, and I'm on a tab that says Graph, and this is the Brainworks web application. There's a couple of things here. There's a home page, a sign-in, which I'll take you through in a minute, and there is a knowledge graph for things surrounding COVID-19. I can zoom in and zoom out of the knowledge graph in this web application. I can click a node to see what it's attached to. I can click one node and click to another node to get a trace of the knowledge that unites or connects two ideas. I can do all sorts of other things too, like I can get the nodes to move through a physics simulation, as you see here, that will help organize the knowledge in a way that it's easier to see. And I can even upload, download data, or control other settings of this graph, all built using exactly the tools that you learned about in this class, including the ability to visualize changes in the connectivity structure for COVID-19 related knowledge over time. You're seeing from 2020 here to 2021. Okay. This whole tool that I showed you and everything that we described, the login and sign up system that you see on the main page, the home page that you see here, all of this was built using HTML, CSS, JavaScript, as well as Flask and a MySQL database on the back. So what you've learned in the class today will allow you to accomplish everything that you saw me take you through in that lecture for the project we delivered for National Institutes of Health, as well as to build something like you see here. And in fact, if you're interested in this, I wanted to note that the code for this particular project is actually available on GitHub. So you can come here to my GitHub page, and you can check out, let me zoom in a little for you, and you can check out the software release for the Brainworks application that was authored by me. This figure should look familiar to you. I covered this in the lecture, right? It's our objective, to structure the scientific literature as that interactive knowledge graph. And we take you through in this repository some information about the need, the solution, the innovation, and then of course the technology stack that was used. So we have the stuff that collects the data, we have the stuff that does the algorithms, and then we have finally the web application that was built for the visualization. So if you actually wanted to see how to build that visualization tool that I showed you, you could see that it uses Flask with your own eyes and check out the JavaScript. You can gain access to it here. You could come to the visualization layer. We've got some examples of how to host your own graph API using the code that we've made publicly available in this repository, using all the tools that we've discussed in class already. And so I wanted to kind of show you all of this with the hope being that you'd be excited about the kinds of opportunities that are unlocked and the cool things that you can do if you've mastered web application development. So with that, I want to welcome you all back from your spring break. I hope that some of this was interesting. If you have an interest in developing full stack web applications, particularly if you're interested in applications that have machine learning in the background, I'd really actually love for you to reach out to me and see if there are ways for us to work together this summer or later in the year.